The next trial to make big news was for patients who are estrogen receptor positive or those with triple-negative breast cancer. This was a phase 2 trial evaluating HER3-DXd, a new antibody-drug conjugate treatment in metastatic breast cancer. If you remember from our first episode, antibodies are sticky proteins that help us to target specific cancer cells while attempting to spare most of the normal non-cancerous breast cancer cells in the body. You've also likely heard of HER2, which is one of the ways that we categorize breast cancers. But researchers are also interested in targeting HER3, which is a similar biomarker found in 20 to 40% of all breast cancers across subtypes, and HER3 is associated with poor outcomes. In this trial, 60 patients with estrogen receptor positive or triple-negative breast cancer received the HER3-DXd drug for about 5 months. Here's more on the outcomes of this study and what we can expect from HER3-DXd in the future from Dr. Erica Hamilton, who presented this study.
We presented results from the HER3-DXd or patritumab deruxtecan at ASCO this year in 2023. In this first 60 patients that we enrolled, the overall response rate was 35%, which was really quite encouraging in patients that had seen an average of 3 prior regimens. And we really wanted to figure out whether HER3 expression mattered for response rates and also how patients with kind of traditionally classified triple negative or hormone receptor positive disease did on this drug. The main takeaways from our data with patritumab deruxtecan or HER3-DXd really was that overall response rate and clinical benefit rate looked encouraging across both hormone receptor positive and triple negative breast cancer. One of the big questions that we really sought to answer, and is a very big topic in antibody drug conjugates right now, is does how much antigen expression a cancer cell has matter? Meaning, what's the threshold for HER2 expression? We know TROP2 we don't measure to give a drug like sacituzumab govitecan. And what we found for HER3 was similar, that patients that had HER3 low expression or HER3 negative expression did no worse than those patients that had HER3 high expression. So really telling us that we probably don't need to measure HER3 expression to determine who may be eligible for this drug. So where we're going from here is we're actively enrolling in Part B and Part Z. Part B is going to enroll a cohort of patients that have triple negative breast cancers and a cohort of patients that have hormone receptor positive breast cancers, irrespective of HER3 expression. And we'll also specifically be looking at patients that have already seen a different antibody drug conjugate. So getting at that question of resistance or sequencing one antibody drug conjugate after the other. And so these patients will have seen trastuzumab deruxtecan, our HER2 targeting agent, or sacituzumab govitecan, our TROP2 targeting agent. And then in Part Z, we're enrolling patients that have HER2 positive breast cancer, and we're enrolling patients that have already seen trastuzumab deruxtecan. So really, in the end of this study, we're really getting at resistance and sequencing and what's the activity of antibody drug conjugate after a different antibody drug conjugate.
Thank you very much to Dr. Hamilton. For more information on antibody drug conjugates, check out a clip from the previous episode of Breast Cancer Breakthroughs, where Komen scholar Dr. Sarah Tolaney describes how antibody drug conjugates work, and we've linked this in the description below.